So most of the time, a single omics, so measuring only gene expression by RNA sequencing, measuring only epigenetics, or measuring only phenotype is not sufficient to get a real understanding of how cell works. So the best solution is to try to combine all of the solution in a comprehensive manner, just to get an understanding of how all these events happen from the nucleus to the external of the cell. And in particular, COVID has been one of those examples where, for example, getting one single omic or a single view of how the system works is not effective to understand how COVID behaviors occurs in population or in clinical hospitalized patients. So we try to combine the genome information and the patient outcome to get the best result of COVID infection. So sequencing the COVID genome was an effective, tedious and laborious procedure. So the main goal of our work was first to make this approach economically affordable, not just for us, but also create a proof of principle by which this approach could become a cost-effective method for any country. So the second approach was to integrate the genome information and the transcriptomic profiling of the patient airway epithelia, just to know not only how the genome evolves and track its evolution, but also to see the response of the host respiratory epithelium. We've used for MGI mostly the COVID solution and the whole genome solution. We are really thrilled to be able to try to test the stereo-seq that I have in production this month. And my point about MGI is that it offers just you an extra alternative solution for your genome sequencing needs. We know that nowadays many sequencing genomic companies are coming up with different solutions. Each one of them, a perfect genomic solution doesn't exist. The perfect genomic solution is the one that better fits with your needs. And with our experience, for example, with COVID, MGI had the right solution at the right moment for our needs, for example, for COVID sequencing. When your focus is not identifying genes or mapping gene expression, but mostly identifying variants or qualifying gene variants like we can have in viral genetics or in medical genetics, it's crucial that none of your sequencing has a sequencing issue, because the sequencing issue might just be an error in the sequencing and misinterpret data. So the use of MGI technology has a huge advantage. So the error rate of MGI technology on DNB sequencing is extremely low. So you can actually rely on the data at the level of best leader in the field, if not better for some applications. So that's what we look for when we start COVID genome sequencing. I'm not a virologist by training, so I had to learn a lot, as many of us scientists, when we try to face COVID pandemic and try to help with all of our skills. We put in the system our knowledge in medical genetics and variants interpretation, and the crosstalk we had with virology, with MGI scientists and genomic specialists, was actually to get the best solution and the best effort to try to get those results as soon as possible, because we've heard and we know that getting results as soon as possible for COVID sequencing is crucial. I'm of course continuing my basic research in my lab, so trying to study how pluripotent cells and stem cell can be manipulated and organized for medical purposes, but we want to use the knowledge that we accumulated in the COVID pandemic to apply the fast, cost-effective and reliable genome sequencing to other types of screening. So we have in mind, for example, to screen for several hereditary cancer, for example, breast cancer, inheritance. And we can effectively use the COVID strategies that we set up for COVID sequencing as a proof of principle to apply the sequencing to human gene and human disease-driving genes.